All right, so chapter 6 deals with changes in phenotypes in populations, so how phenotypes evolve. And we're going to start with the foundational understanding that we've established of the genetic basis of phenotypic traits and how those genetically based phenotypic traits are passed from parent to offspring, and how the variation that drives that the genetic variation that drives that phenotypic variation arises. It's something we've learned about and talked about, mutation. So first I want to introduce a term to you, and that is quantitative traits. So a trait that has quantifiable variation. But here we can see an instance of a quantitative trait, that being height in humans, that exhibits continuous variation over a range of sizes, right? So there are, in this group of, it looks like military personnel, there are individuals grouped by height. So everything from 5 foot tall to 6'5", right? And what's interesting is you see that there's almost a normal distribution, right? Like a bell curve, where most of the individuals fall somewhere between 5'6" and 5'10", and there are very few at the 5 foot range, very few at the 6'5" range. One of the things I want to make clear is that there's not discrete size. There are not discrete sizes within this, right? So the people that are 5'9" are not all exactly the same height, right? Even between 5'8" and 5'10", there's a gradient of variation that's far more discrete than the 1 inch increments that we're sorting individuals by in this image. So these traits, this variation within this trait, height, is passed on by individuals. So you can imagine that if 2 individuals from that 5'8" column had offspring, their offspring would probably be around 5'8", right? But it's not necessarily true, right? Shorter, both of my parents were right around 5'9", 5'10", and I'm nearly 6'4". So there are some interesting combinations that happen with these traits that are controlled by multiple genes, as height is, right? So there's not just one gene for height that has only 2 alleles, right? There are many genes that contribute to the height of an individual. And so the inheritance of that complex suite of alleles for multiple genes can lead to highly variable populations like this, in which the trait exhibits very discrete variation. So this figure from your text illustrates some of these principles, and it also brings into the fold the influence of the environment. Okay, so going from top to bottom, we are looking at traits that exhibit some sort of variation, and they are traits that are influenced by 1, 4, or 10 different loci or genes in the genome. As you remember, with one locus, if there are 2 alleles, there are going to be 3 genotypes. And you can see in these top graphs, there are 3 blue bars, each of those representing the trait value on the x-axis for each of those 3 genotypes. And so you can imagine that the homozygous recessive, let's say, exhibits a trait value of 8. This could be size. Let's just say it's 8 millimeters long. Heterozygotes are 10 millimeters. Homozygous dominant individuals are 12 millimeters, right? I'm just making up the genotypes to match this, but you can see those 3 different genotypes and their trait values. Now, the difference between the graph on the left and the graph on the right is the width of those green lines, okay? Now, the green lines are taking into account environmental contribution to that phenotypic trait value, right? So look at the top left graph. So that the green line around the trait value of 10 is very narrow. And that means individuals that are, say, heterozygous, in this example, exhibit trait values somewhere between 9 and 11, right? Depending on the environment. And the environment cannot exert or allow even greater variation in that trait value in that circumstance, if the environment doesn't contribute much. On the other hand, if the environment does contribute a great deal to variance around that average trait value, let's say size, We go over to the graph on the right. Okay, so the trait value for heterozygotes is on average 10. Okay, but if the environment contributes greatly to that trait, then there can be extremes even with that same genotype. Individuals could be 6, could be 14 with that same heterozygous genotype if there's a significant contribution of the environment to the phenotype, right? And you can think of instances where the genetics and the environment play a role in phenotype, and one is human body type, right? So genetics and your environment contribute to levels of obesity in humans, for example, right? I might be genetically predisposed to be slim, but if environmentally, all I have to eat is crappy prepackaged food that's high in fat and high in calories and low in nutritional value, I might be, despite the fact I'm genetically predisposed to being slender, I might fall somewhere else on the spectrum of obesity. Okay. So as we look further down in this figure, as you add loci contributing to this phenotype, you can see that you get a much broader, more evenly distributed pattern of phenotypic variation within the population. So the more genes contribute to something and the more influence the environment has on the phenotype, the more gradual the variation will be across the population. Okay. Now, what's really interesting is when we start looking at examples later on in this lecture where selection is acting on traits that are influenced by multiple loci, and we can start to see some really extreme trait values arise after selection. Now, when we think about variation within a trait, we also want to think about the fitness of those different trait values, because not all different phenotypes within a population have the same fitness, as you might suspect. So here's an example of a horned lizard, and on the bottom left, and you can see on the right a horned lizard that has been caught by a bird, this bird being a loggerhead shrike. Okay, these shrikes are highly predatory, small birds that will eat these horned toads, which are actually lizards. And they do this by impaling them on barbed wire or spines of trees, as you can see in the top left. So they go catch these horned lizards, impale them, and that allows them to eat what they can from the lizard's body. Okay. Now, these horned lizards have these horns for a reason. They do serve to protect them. And it turns out the larger those horns, the harder it is for the shrike to catch them and impale them and consume them. So the longer the horns are, the better your survival. And so we can see the relative horn length in the graph on the top right, where the shorter the horns, the lower the survival rate. Larger horns, higher survival rate. Okay, so this gives you an illustration of how fitness acts on a continuously variable trait, right? We're no longer thinking about homozygous recessive, heterozygote, and homozygous dominant genotypes and their relative fitness to one another. We are looking at a continuously variable trait, how long the horns are, and what the survival rate is for all of those different horn lengths. Longer is high, but you get below some threshold and there's a pretty rapid drop off in survival rates. So if we think about these variable populations that have extreme trait values occurring at, let's say, some low frequency and average trait values at a high frequency, like all of the top blue graphs in this figure, we're looking again at these sort of normal distributions of how, so the y-axis is frequency. What is the frequency of individuals in the population with any trait value on the x-axis? And we're starting out with what we call kind of normal distributions, high in the middle, low on the sides, okay, low to the left, low to the right, the frequency that being. And each of these populations being subject to different types of selection, okay? And the selection pressure is illustrated and related to the trait value in the middle series of graphs. And you can see here that survival rates are very different under these different scenarios of selection, whether selection be directional, stabilizing, or disruptive. And what we're seeing in this graph, we're going to work through each of the three columns here separately, is that if you start out with the graph at the top and you subject that population to the selection regime illustrated by the red line in the middle, what you're going to end up with is a new population after selection that's on the bottom of the series of graphs. So let's first start with directional selection. And this is probably the most common form of selection, the one we think of most commonly, right? Selection for deer to be faster, for fish to be larger, for lions to be stronger, right? Selection pushing the population towards an extreme. That's what we're talking about with directional selection. On the left, you can see a population where the red diamond indicates the average trait value for something, right? Somewhere in the middle. It is the most frequent trait value in the population. Now, if survival is lowest at low trait values towards 0 and highest at high trait values, right? You get that diagonal line of survival across all trait values. What that means is individuals on the left side of the x-axis having very low trait values are having very low survival. They are not going to pass their alleles on to the next generation. And so the individuals that are breeding are those that survive, that have on average higher trait values and pass those high trait values on to their offspring. And so what we see in the later population, the bottom left graph, is that trait value has shifted a bit. It's now somewhere right between 5 and 10. So let's say it's right around 7.5. Whereas in the initial population before selection, it was about 6. So selection has pushed the population in one direction towards larger trait values. An example from the text is on the right. This is a population of finches where the number of individuals that have any certain beak depth, right? That's from top to bottom. How chunky is that beak? Okay. So you can see that there were very few individuals with very shallow beaks, let's say 7 millimeters. And there were very few individuals with really deep beaks, let's say 11 or 12 millimeters. And most individuals had an average of, let's just call it 9.5 millimeters or 9.75 millimeters. Okay. Now, when selection acted on this population, there was actually a drought. And look at the survival rate in the top right. This line approximates a bit the middle red line off to the left in the directional selection example. You can see that survival is very low for individuals with shallow bill depths, right? Below 9 millimeters, low survival. So basically what you're doing is taking that middle finch graph, and anything below 9 millimeters didn't survive, didn't pass on their beak size alleles to their offspring. But survival is incredibly high to the right side of the size spectrum, so that individuals with 10 and 11 and 12 millimeter beaks actually had very high survival, and they passed those alleles on to their offspring. And so before and after selection, what we are seeing is a shift in the average size of the beak of these finches in the population to the right. They are getting larger and larger, these beaks of these finches. Human populations, on the other hand, have been under stabilizing selection for birth weight. Okay, so let's first look at the three graphs on the far left. We have a population with an average birth weight size, right? And that average is somewhere in the neighborhood of 7 pounds, okay? Now, some individuals are born at very low birth weights, let's say 1 pound, and some are born at 10, 11, 12 pounds, okay? Selection sees low survival at very low birth weights, low survival also at very high birth weights, right? Both of those present problems for birthing, and survival is lower. So survival is highest at that average trait value. What selection is doing is stabilizing the population for a size distribution close to the average. Average is best, okay? Extremes have lower survival and lower fitness. And so what happens under stabilizing selection is the population stabilizes around some optimal average value. And you can see that in the bottom graph, okay? We are removing extremes from the population and the distribution becomes narrower and narrower. If you look at the graph in the middle next to that baby on the scale, you can see that survival rates are very much decreased. So follow this blue line. Survival rates are decreased on the lower end of the size spectrum dramatically. And they also begin to decrease as you get above 10 pounds. And so for that reason, human birth weight is said to be under stabilizing selection, balancing the population around this midline optimum. The third form of selection acting on phenotypic variation within a population is disruptive selection. Okay. And in this is the opposite of stabilizing selection. And indeed, you can see the red line in the middle on the left is actually an inverted version of what we just saw in stabilizing selection. And what that means is that individuals that have average trait values or are in the middle of that phenotypic distribution in the top blue graph there on the left, those average middle individuals have the lowest survival. The highest survival is at the extremes of the population variation, okay? Hence, we have a red U-shape selection curve. And what that means is individuals at both extremes have the highest survival, and we are going to see the population diverge to have much smaller and much larger sizes. In the example presented on the right, this is a species of bird, a crossbill, that feeds on the fruits of these coniferous trees, both hemlock and lodgepole pines in the mountain west of the U.S. And so beak size is optimized for eating either these really large lodgepole pine cones or these much smaller hemlock cones. And there is nothing in the middle. So if you have a medium-sized beak, it's too small to eat a lodgepole pine cone. It's too big to effectively eat a hemlock pine cone, okay? So those intermediate values have the lowest survival, okay? And so the frequency of these different beak sizes will change over time, okay? So in the example in your text, they talk about these populations that have either an average illustrated by peak 1, 2, or 3. Peak 3 is fine, right? If that's the average of a population and they have access to lodgepole pines, they're fine. And I'm sorry if my head is covering peak 3. It's the rightmost light blue curve on the bottom right of the screen. Peak 2, suboptimal, right? That population can't eat either hemlock or lodgepole and is going to shift left or right. It's average beak depth. The population number 1 is slightly too small to actually eat the hemlock cones. And so they are likely to shift a bit to the right to get under that ideal beak size to consume hemlock cones. Okay. So this is a scenario where these birds are under disruptive selection. Now, fitness can depend on multiple components of the phenotype of an organism. And remember, I've cautioned you to realize that the phenotype is not just what you see in an individual. So in this case, we're looking at a garter snake that has a bright yellow stripe right down the middle of its back, right? That's something I can see. But another component of the fitness of this species of snake is its behavior, right? The combination of that color and how it behaves determines the fitness of an individual. And your text has just a fascinating example that's illustrated in a graph that's a little tough to decipher if you've not looked at a three-dimensional graph like this in the past. But I encourage you to think of the three-dimensional surface of this graph like a sheet of paper, right? And so it's that the edges of that sheet of paper are demarcated by the really dark green lines here. And so you can see that two corners are pulled up high, and two corners of that paper are kind of pulled down low. So you could kind of twist that paper, take a sheet of paper and twist it, and you could approximate the surface of this graphed fitness landscape, is what we call it. So I want you to think about what's being illustrated here, okay? In these populations of garter snakes, there are actually two different phenotypes. The phenotype of having a bright stripe down the back, like I showed you, okay? And the phenotype of being spotted. So garter snakes in these populations can be either or some, mix of combination. So you'll notice that the y-axis is fitness, okay? And the axis that comes right off of that is stripedness. How striped are they? Further to the back, towards point 1, they're pretty striped, like that orange striped snake on the top left there. As you come closer to use, if stripeness gets down to -1.1, that is a very unstriped individual, okay? It's a very spotted individual. Now, the other component of this axis is the tendency to reverse course when fleeing, okay? And so I want you to think about a snake trying to get away from a predator. It can do a couple things. It can take off in a straight line, or it can start, stop, reverse course, change direction, and move around in a very, distracting type of way, okay? So we've got those two components of the phenotype, right? One is, are they striped or not, striped or spotted? The other is, how do they try to escape that behavior? And again, that behavior is a part of the phenotype of these individuals. It's genetically encoded. And it turns out there are optimal combinations, right? You take that piece of paper and twist it. Two of those corners are higher than the other two. Those two peaks on the fitness landscape, the highest peaks, are the most ideal combinations of coloration and behavior. And it turns out if you are on the left side of the x-axis, your tendency to reverse course when fleeing is high, right? Come way over to the left towards that 3.5, okay? If you have a high tendency to reverse course when fleeing, okay, the highest point is the one that's closest in terms of stripedness, not striped. They're spotted. So spotted snakes that reverse course when fleeing, that's the corner of this graph that's closest to you, and that's the highest point, okay? The other high point is what I would call the back right corner of this graph, okay? Far to the back, that means they're very striped. Far to the right, that means they do not reverse course when fleeing. I want you to think about this snake in the grass, and if you are watching this snake as a predator and it is just going in a straight line, the really hard thing to do is tell when the snake is actually moving. It might look like it's just sitting there because that stripe, you can't tell it's moving. And then all of a sudden you as a predator land on it and it's already disappeared because it was hauling ass and you didn't know it. Because the stripe going in a straight line makes it difficult to assess how fast it's moving. So that's an optimal combination. Those two combinations represent the highest peaks of this fitness landscape. And so there are instances where multiple components of the phenotype contribute to the fitness of an individual, not just how deep is the beak or how much does the baby weigh, okay? So there are ways in which we can determine the strength of selection acting on a trait. Here is a measure or an analysis of the fitness of male guppies with the x-axis representing how orange they are and the y-axis their relative fitness. So the more orange you are, the higher the fitness is, right? So this is a quantification of the strength or the selection gradient that exists within this population of guppies. So this gradient can range from a very low difference in the strength of selection or a very low power of selection to extremely strong selection gradient, right? So if that selection gradient is, somewhere below 0, okay, that's very uncommon. This red line illustrates how common are various selection gradients that have been quantified and observed in nature. You can see most of them are right around 0. They're not real strong. They're not real positively strong. They're not real negatively strong, right? They're not going to push the population real strong in one direction or the other. Okay. That distribution of observed selection gradients decreases rapidly, but goes out pretty far. So that means way out towards -1, there are some instances of very strong negative selection gradients. And far to the right, there are some instances of very strong positive selection gradients. Okay. And each of the graphs at the top illustrates one of these 3 instances. Okay. You can see at -0.5 and +0.5, same strength of selection, but they're pushing the population in 2 different ways. Look up at those first 2 graphs at the top. The -0.5 population, you are going to see an original green parental population selected on directionally to the left to change its size, smaller, and the resulting population of offspring will have an average that is slightly smaller, that blue bell curve. Okay, so we've seen a small shift in the average size of the population to a smaller size. The middle graph shows the same strength of selection, but in the other direction, also directional selection, we see a shift to a slightly larger population of offspring than the parents. Now, if selection is much stronger, go out to the positive selection of 1. Look at that graph. We see the same thing where the offspring population, that blue bell curve, is shifted to the right, but the magnitude of that shift is greater because the selection gradient was larger. Okay. But what I want you to take away from this is that most selection within populations is not all that strong, right? The frequency is much highest around the peak of that red line. But there are instances where selection is extremely strong in both directions. So let's think about quantifying how strong selection is and what the result is going to be. Okay. We've got some graphs on the right that I really want you to make sure that you understand. Okay. And if you understand these, you can run through a calculation that we call the Breeder's equation very quickly and make predictions about what's going to happen in a population, right? So on the right, top right, we're looking at a population where there's a very high correlation between the average of the parents on the x-axis and the average of the offspring on the y-axis. We've seen this same graph before. You've seen the second graph before with selection, right? Where parents to the left of that dashed line don't reproduce, so they produce no offspring, and only the individuals to the right of the dashed line produce offspring. And so what I want to make clear here in graph B is that the parents that survive and reproduce in this graph, so it's Z bar with the little asterisk or star, so Z bar star is the average of those parents that survive, okay? And because there's such a tight relationship between the size of the parents and the size of the offspring, okay, the average of the offspring is here going to equal Z bar star, okay? Now, what I have not spoken to you about is the fact that the slope of this line, it represents how heritable a particular trait is. If the slope of that line is 1, right, an increase in 1 unit on the x-axis results in an increase of 1 unit on the y-axis, there's a perfect 1-to-1 relationship between the average of parents and the average of offspring. That would be a highly heritable trait. And so the slope of this line is heritability, okay, indicated by h squared. So the slope of the line in this instance is very high, right around 1. So we see a very close relationship between a change in the parental population and a change in the population of the offspring. What we are going to do now is dig into how we can use our understanding of the heritability of a trait and the average of the parents in order to make predictions about changes in the population later on, focusing on graph C here. Okay, so let me just show you one example from your text in which they observed heritability of beak size in finches in 2 different years. Okay. And what they found is that the average of the parent's beak size was very predictive of the average of the offspring beak size, right? So heritability was calculated to be quite high, somewhere in the neighborhood of 0.9. Okay. So basically, the parent's beak size predicts about 90% of the offspring beak size. Now let's look to the left and see what the difference in highly heritable versus non-heritable traits looks like. Okay. If a trait is highly heritable, heritability can reach 1. That's a maximum value of heritability. Okay. It can't be, that's a perfect fit between parents and offspring. It's 100% predictable. Okay. The exact change in the offspring. If heritability is 0, that top left graph, you can see it doesn't matter what the parents are. Okay. The offspring can be anything. And so there's very little relationship between parental average and offspring average. And usually phenotypic traits fall somewhere in between these two, right? A heritability of 1 is incredibly uncommon, right? There's some variation introduced by other aspects of the environment and whatnot. Heritability of 0 could be something, as simple as, right, your taste in music is probably not highly heritable, right? But your affinity for chocolate might be a bit more heritable, okay? So let's look at how we can use our understanding of heritability to make predictions about subsequent generations. So if we think about a population in which there's a parental average down on the bottom on the x-axis, z-bar, and if all of those individuals in the population bred and had offspring, then the offspring would have an average of z-bar, right? If heritability is 1, as we've got illustrated here, the parental average and the offspring average is going to be the same. But what's illustrated by S is selection acting on the population. Remember when we had that graph where it showed individuals to the left of that dashed line expiring? They didn't survive, okay? What that means is the parental average shifted due to selection to z-bar star, okay? And with this heritability, we can track if z-bar star is a certain number, then z-bar prime, the new predicted average for the offspring, is going to be something that is reflected by the heritability. So the heritability has determined how high we go up on the y-axis, and S has determined how far to the right on the x-axis we go. Okay, so let's work through an example with the understanding that selection is this difference. So S is a number. It represents the difference between z-bar and z-bar star. It is the difference between the average of the parental population, z-bar, and the average of those that actually produce offspring. That's z-bar star, right? That difference is important, okay? And with that difference and an understanding of heritability, we can make a prediction about what z-bar prime is and what the change in z-bar is. So delta z-bar. Okay, so let's just imagine a scenario where on your farm you have 100 dairy cows, and these 100 dairy cows produce on average 4.8 liters of milk a day, right? That number, 4.8 liters, represents z-bar on the x-axis. That's the average of the population. But you don't need all of your 100 dairy cows to produce offspring, and you want to selectively choose those that produce offspring so you get more dairy, you get more milk, right? So you choose to breed only those that produce 5.2 or more liters of milk a day. And if you take all of those individuals from 5.2, and maybe you've got one that produces 8 liters of milk a day, right? Take all of them, they average 6 liters of milk a day in production. That's z-bar star, okay? So your difference between Z bar and Z bar star is the difference between 4.8 and 6 liters, okay, the two averages. And I'm giving you the heritability of milk production. This is a highly heritable trait, so 0.85 is your heritability. So if you look up that equation in the top left, you can see that with heritability and the strength of selection, S, we can make a prediction about what that change in Z bar is, what delta Z bar is. And with that, we can figure out what Z bar prime is, okay? So this difference of 1.2 liters of milk a day, S, multiplied by the heritability 0.85, gives you a 1.02 liter difference or increase in milk production in your offspring, okay? So that delta Z bar is 1.02. So if I take an increase in 1.02 liters a day over our previous production average of 4.8, I get 5.82 liters of milk a day in my next generation. That's a really rapid shift in milk production that is very predictable based on the strength of selection that I, as the dairy cow breeder, get to choose. Okay, so shifting gears a little bit, we're going to dig into a bit more of the examples of exceptions to these very simple single locus, two allele models that we've described. There are a few things I want you to understand when it comes to selection acting on variation within populations and the traits that are being acted on. And we're going to start digging into these instances where there are more than one loci contributing to the phenotype of an individual. The first is a term that you've probably seen before, epistasis. This is when multiple genes at different loci, so different genes, interact with one another on a single phenotypic trait. The best example that most folks in the Southeast are familiar with is the coloration of Labrador retrievers that can be black, chocolate, or yellow in general. So there's a gene that determines the color that is deposited in the hair follicles, right? And that color can either be black or brown, right? So if the dominant allele is black, so if you've got a big B in this case, then the puppy is going to be black, okay? And if you are homozygous recessive for B, that produces only brown coloration. So chocolate labs are homozygous recessive for the black color allele, so they are brown. Black labs can be either homozygous dominant or heterozygous. One copy of that dominant black allele, you get black hair. Now, the epistatic gene, E, is one that determines, the product of that gene determines whether that pigment that is produced is actually put in the hair. So there's a gene that makes the pigment, then there's another one that puts it in the hair. If a dog is homozygous recessive, little e, for that gene product that puts the pigment in the hair, then you get a yellow lab. And that yellow lab could have a big B or not. Could be homozygous dominant for big B. It could be homozygous recessive for B. Okay, doesn't matter. No matter what pigment is being produced, it's homozygous recessive for that E locus. It doesn't put any pigment in the hair of the dog. Okay, so because of this, we have two different genes whose alleles are contributing together to determine the phenotype. All right. So this is an example of very separate unlinked genes that could be selected upon in tandem. Okay. Now, as new mutations are arising in the population and the population is adapting to new environments, right? We've talked about the role of these populations being very large. So why is it that large populations are important? Why do biologists become concerned when populations of endangered species get smaller and smaller, right? So it has everything to do with all of the mutations that have arisen in the population, the genetic variation that exists within the population. Necessarily, a large population will be able to hold and have more genetic variation than a small population. So they are generally believed to be healthier and better able to respond to changes in the environment, right? This is a very large population of penguins in this photograph, okay? And so presumably there's a great deal of genetic variation within this population, okay? But if you have this understanding of the mechanism whereby mutations arise, and those mutations are the things, the fuel that drive evolution, that allow evolution to happen because they present varying phenotypes within the population, some of which may be advantageous, some disadvantageous. You'll have a better understanding of how these populations respond to changing environments. And so the text goes into explaining why population size being large and or having lots of genetic variation in a population is critical to the survival of a population. And so here we have an example of, let's first consider the blue line, where a population is of some average size and does not have a whole lot of genetic variation, right? It's not huge, but it's not small, but it does not have a lot of genetic variation. If the population size, if the population is subject to some environmental change, so if something changes at that dashed line in time, the population declines to the point where it gets so small that it could and will likely go extinct. It was already not a huge population. It didn't have much genetic variation. It could not adapt to the changes in the environment. The red line, on the other hand, a very large population with similar genetic variation to the blue line population, it also decreased in population size very dramatically after the environment changed. But because it was so large, the population was large enough to have some genetic variation that either arose or existed within the population, and the population can survive, okay, and increase in size again. The green line represents a medium-sized population that for some reason has an enormous amount of genetic variance. Lots of genetic variation already there. And so that dip in the population size was much less pronounced than in the large population that did not have as much genetic variance. Okay? So the response of populations to changes, this is a change that affords different fitness levels to different genotypes and phenotypes. The response of that population is entirely dependent on how much variation there is in the genotype, in the phenotype of these populations. Okay, so that's why biologists become concerned about endangered species, because their populations are small. They necessarily have very little genetic variation. And if, let's say, a virus came through and started wiping out individual bald eagles, right? If the population is very small, then it could be that there aren't enough of them that there exists individuals that are resistant to this virus, and the whole species could go extinct. The larger the population, the greater the variation, the greater the ability to respond to environmental change. Okay, and this is an experimental population of nematodes in the lab. They're a model organism that geneticists study, and they can grow millions of these worms on plates, like bacterial plates almost. And this is a population that was quite large, okay, high density of individuals, that the population was subjected to exposure to salt, which killed a lot of individuals. This is a logarithmic scale, so you can see that cell density, basically the abundance of these single-celled worms on the plate, went from about 100 down to somewhere in the neighborhood of 10. So a 90% reduction in the population size, okay? But they were able to come out of it and increase in size, in the population size, because there were enough individuals in that population at the beginning that mutations had arisen, allowing them to survive, right? And by the time we get to the trough of this red dip, what has happened is only those individuals with the mutations that make them able to survive in salt are there having offspring. So it's 10% of the original population, and then they just begin having offspring that are all resistant. Okay, so the population grows again down the road. And we can learn a lot about these scenarios of populations evolving over time due to selection, not just using nematodes in the lab, but also observing domesticated organisms and how they have changed over hundreds of years that humans have worked with them. So if you go out and get a pizza, right, you are consuming a number of domesticated species that humans have artificially selected for over many, many thousands of years, right? Including wheat, tomato, and dairy-producing cows, right? And you can see the difference between the wild ancestors here and what we are currently growing as domesticated species. And these are still getting bigger and better, right? If you want to think about it that way, right? The text mentions that dairy production from cows continues to increase by 1% to 2% a year. That's incredible. That's an enormous increase in dairy output from these cows. So another example that we see quite frequently are purebred dogs that have been selected for very specific traits. So this bulldog has a very specific, short, muscular physique that is quite distinctive, right? And the only way that we dog breeders are able to maintain these consistent phenotypic traits is through preventing the mixing of individuals that would cause their offspring to share the traits of these mixed parents, right? So that's why there's for folks that are really into purebred dogs, they are very careful about making sure that their poodles breed with poodles. Now, these traits that are evolving and are under selection, interestingly, can evolve beyond the original range of variation that existed, right? So think back to that first slide in this lecture where I showed you basically a population of humans that ranged from 5 foot tall to 6'5", right? If we sent that population to Mars, all those individuals went to Mars and they founded a new colony, and it were beneficial to be taller, or let's make it shorter. It's probably better to be shorter on Mars, okay? It's better to be shorter on Mars. I bet if you go back to Mars in a couple thousand years, the shortest person is not going to be 5 foot, right? They're going to be people that are 4'6" on Mars. Even though that is outside the range of variation of the population today, through selection on these traits that are determined by many, many genes, we can find new optimal combinations of alleles that exist today that produce new phenotypes that are more optimal. If you look at the last 100 generations of corn that have been grown here in the United States, you can see that the oil content of corn used to be quite low. 5% of the volume of that corn could be extracted for oil, right? For biofuels or for cooking oils. But you can see over the last 100 years, selection has increased that oil content dramatically to the point where corn today is more than 20% oil content, right? That is a far cry from the original, 100 generations ago, where it ranged from 4 to 6%, let's say. Oil content today is not entirely due to brand new mutations that have arisen. It is not due to genetically modifying the corn that we are growing. It is due to selecting and breeding those individuals that have specific traits, high oil content, so their offspring will have even more. And then taking those offspring and breeding them so that there will be even more oil content. Okay. Now, coming back to this influence of population size and the rate at which populations can evolve, Here are 3 different fly populations in which selection was imposed upon these populations for longer wings, okay? And so the control population had a certain wing size, right? And so what we see is over 60 generations, if we compare the control population to the population that we experimentally selected, we can see that the experimental populations increased in wing size relative to the control pretty significantly, right? Now, there are 3 different lines here for 3 different populations, one of 1,000 individuals, one of 200, and one of 40. You can see that the larger the population is, the more rapid the increase in wing length is. The population is responding faster to selection because it is larger, okay? And what you need to know about this experiment is that for each generation, the scientists would breed a bunch of fruit flies, and then they would pick the 200 individuals that had the longest wings and use them to make the next generation, okay? Or they would take, they would take the individuals with the longest wings and use them to breed the next generation, okay? Let it get up to 1,000 or 200 or 40, and then take another batch of the longest wings for the next generation, okay? The bigger the population is allowed to be, the more rapid the change in the population can be, okay? A small population of 40 flies can't evolve as rapidly, even if the strength of selection is the same, can't evolve as rapidly as a large population of 1,000 individuals. Now, when selection is acting on these traits that confer some advantage, okay, then there can be negative consequences, or there can be positive consequences that come along with it, right? But when we think about these dairy cows and selection acting on them for increased milk production, there is, in fact, a negative byproduct of this selection. And that actually results in, the fact that we're getting more milk out of these cows. Turns out the cows are less and less fertile, okay? So there's a balance. You're getting one benefit, but there's also a detrimental aspect to that selection regime. So the direct response is this increase in milk production. The indirect response to selection is a decrease in fertility, right? Remember that example of selection on snail shells from our previous lecture, right? Selection acted on whether the shells were striped or not. So the direct response to that selection was that all the descendants were not striped. The indirect response was that they were also pink, because only there were no unstriped yellow shells. So there can be these correlations between different traits, okay? So if there is no such correlation between trait 1 and trait 2, right, then if we have a population on the top left before selection with some average, okay, and selection acts on trait 1, okay, the average for trait 1 shifts to the right, but it has no effect on trait 2, up or down on the y-axis, okay, because there's no correlation. There's no correlation between trait 1 and trait 2. Conversely, if there is a correlation between trait 1 and trait 2, okay, selection for trait 1 results also in a change in trait 2. So let me give you an example of a genetic correlation, okay? What if, let's imagine that, selection acts on humans for arm length, right? Individuals with really long arms survive. Well, if you compare arm length and leg length, right, they are highly correlated, right? So despite the fact it's longer arms that are helping you live, individuals are also going to have longer legs, okay? So there's an example of a genetic correlation. Selection acting only on one of the traits, but resulting in a change in both of them. Now, there are genetic constraints on the way in which selection can change a population. Go back and look at this bottom left graph, right? Because there's a genetic correlation between trait 1 and trait 2, the population lives within this diagonal space of trait values, okay? As does this plant, okay? Where you can see that the leaves on the bottom, the x-axis, and reproductive stage on the y-axis, there's a pretty significant correlation between those two traits, okay? And so genetically, there's what they're calling a line of least resistance, right? If selection's acting on this population, the population could very quickly move up and down this blue line in response to selection. Where the population is going to have a harder time evolving is if evolution is pushing towards, if the optimum trait value is somewhere like that red target dot on the top right. If that's where the population needs to get to, that's a no-go, okay? There's an evolutionary constraint there, that constraint being the genotypic variation and combinations that exist in the population don't allow the population to move on the axis of that red dotted line, okay? The population can move on that blue line, but there are genetic constraints that constrain the evolution of this population from reaching some optimal phenotypic combination, okay? So this is a really important point because we're learning about the power of evolution in organisms evolving to adapt to changing environments. And there's no doubt this has been critical to the survival of millions of species over the history of life on this planet. We hear a lot these days about climate change and changing environments. And no, this isn't the only time the climate has ever changed. That's not the argument. The idea is that the climate's changing faster than it has in the past, but that's a conversation for another day. But recognize that these changes that are happening in the environment, not all populations are going to be able to evolve and adapt to these changes because there are likely to be a number of evolutionary constraints that prevent them from doing so, okay? So these genetic correlations that we've talked about, why might these exist? Okay, so the first is a term that I want you to be familiar with, and this is pleiotropy. So when one locus, a change to one locus, has multiple effects, right? This is kind of the opposite of epistasis, if you want to think about it that way. Remember, two loci affect one trait. This is one locus affecting multiple traits, okay? So mutations to a particular gene, I've talked about an example of pleiotropy with respect to Peter Dinklage, right? And his, the guy from Game of Thrones, and his achondroplasia dwarfism, right? That is a single mutation that has multiple effects. So this pleiotropy is one cause of these genetic correlations. There can also be instances of linkage disequilibrium, also something we've talked about. Notably, the linkage disequilibrium we've spoken about to this point is when genes are linked physically on the same chromosome. That's not the only way it happens, right? There should be a linkage disequilibrium in these snake populations for the color pattern they have and the way that they try to escape. That's another instance of linkage disequilibrium, selection acting on two things in tandem. That is a genetic correlation. So when we think about the phenotype, and we've talked about the phenotype being derived from the genotype. It's the genotype that determines the phenotype, okay? Is the phenotype deterministic? What does that mean? That means, does the genotype determine 100% exactly what the phenotype is going to be? Turns out, I'm going to spend the rest of this lecture, which is not a whole lot more, you're welcome, talking about ways in which the phenotype is not deterministic. And this gets into something we call phenotypic plasticity, okay? And that's pretty much self-explanatory. The phenotype associated with certain genotypes may be plastic, may be flexible, depending on environmental scenarios, okay? So the example your text uses is a very compelling one. This is looking at populations of spadefoot toads, this adorable little frog on the bottom right. This isn't the species we have here in Oxford, but we do have spadefoot toads here in North Mississippi. This is a desert-dwelling species that interestingly lays its eggs, well, necessarily, it lays its eggs in pools of water, little ponds that form in the desert during the rainy season. As you might suspect, the rainy season in the desert isn't all that long. It's not a lot of water that comes down. What's really cool is the first frogs to go lay their eggs in these rain pools or ponds at the beginning of rainy season, their offspring grow up like normal tadpoles and eat algae and other small things swimming around in the pools. Okay? If a pair of spadefoot toads breeds late in the rainy season, just before the pools dry up, their tadpoles don't have much time. Okay? So despite the fact that the genotypes might be the same, okay, the fact that these individuals were born in these ponds very late as they're getting smaller, they don't have enough time to grow on algae, and they are carnivorous and cannibalistic. And so you can see here a big spadefoot toad tadpole eating a smaller spadefoot toad tadpole. Okay, this is a large cannibal eating a smaller herbivore, and it's quite possible that that smaller herbivorous individual is older than the big cannibal, but these big cannibals have to grow rapidly because the pool is quickly drying to nothing. So this is an instance where the environment is influencing the phenotype, okay? And a particular genotype can have two possible outcomes. It can go to be an herbivorous, early born individual, or if they're late born, they can become cannibalistic. So this gets into the interaction between genotype and the environment as a contributor to determining the phenotype of an individual, okay? And this example in your text describes different populations of these small aquatic invertebrates that can either have lots of dark pigments or very little to no dark pigments, right? As you can see on the far right. So dark pigments have a benefit and a cost, right? The benefit is, as these organisms are living near the surface of the water, it protects them from UV light, which damages DNA, okay? So if you're in the water and floating around where these things do, it's best if you don't get sunburnt, okay? So the benefit of melanin protecting them from UV light is quite high, okay? Especially if they are in high light environments, right? Now, if there are predators around, being dark like that makes you very visible in the water column and you get eaten, okay? So there's a consequence to that. So it turns out that populations that don't have any predatory fish, so pools where these things live, if they're exposed to high levels of UV, there's an increase in melanin production, right? So those two purple lines show two populations without predators at different light levels, okay? So the more UV, the more melanin they have. That's not the case in populations where there are lots of predators, okay? There's a slight increase in melanin production, but not as much as if there were no predators, okay? And so this represents a genotype by environment interaction. You see up at the top of the slide, I put GXE. That's genotype by or multiplied by environment interaction. So if there is a difference in the phenotype depending on the environment, right? So any one of these lines, just look at one of these lines. Each one of those lines illustrates a genotype by environment interaction, okay? Any one of these genotypes responds differently under high UV light. Okay, that's the genotype by environment interaction. Now, the fact that the purple lines and the red lines have different slope is illustrating a separate principle entirely, and that is a reaction norm, or a norm of reaction. Okay, so if there are different responses of different genotypes under the same scenarios, okay, the different genotypes being the genotypes that live where there are no predators and the genotypes that live where there are predators, those reaction norms, the differences between those, okay, that's a different scenario whereby genotypes react differently to the same scenarios, that being high light or high UV intensity. So their direct response differs. That's the example of a genotype by environment interaction. Okay, so that's where I'm going to wrap up chapter 6. You can skip this QTL section if you're reading the chapter, as you should. And we will pick up with live synchronous Zooms on Tuesday of next week.